Hello everyone, how are you? I hope good. I'm good as well. Well, today we are going to understand a very important yet difficult concept in biology, which is human digestive system. Now, human digestive system consists of alimentary canal that extends from mouth to anus. So as you can see, I have already made the mind map of a part of human digestive system over here that will facilitate the understanding of this concept. Right, so we'll start from mouth. As you can see, mouth is a part responsible for ingestion. Ingestion is to take food in, right? The food is getting inside. Now, the moment the food is inside, the teeth present, right? The teeth will break down the food particles into small pieces so that the enzyme can act on it efficiently and easily. Then the chewed food basically is inside the buccal cavity or oral cavity. Now, in the oral cavity, there are salivary glands. Can you see these tree-like structures? These are actually the salivary glands that secretes saliva. Now, the pH of the saliva is 6.8, so we can say it is slightly acidic, right? Now, what is saliva contains? Saliva contains very important enzyme, which is salivary amylase. Here, S stands for salivary. Another name is ptyalin. P is silent. So it acts on starch component of the food, and convert it into simpler form, simple sugar, which is maltose. What else does it contain? Saliva contains, it contains lysozyme. Lysozyme, if by chance you have ingested bacteria along with the food, it can help in killing the bacteria. Alright, now this process of chewing food and mixing it with saliva, the process is known as mastication, right? Now the food, the semi-digested food mixed with saliva, lubricated with it, is given a term which is known as bolus. Now the bolus is ready to enter the next part of the alimentary canal, which is esophagus, also known as food pipe, right? Now what's there in esophagus? Esophagus connects the buccal cavity with the stomach. As you can see, this is the esophagus. This is approximately 25 centimeter in length, but of course it can vary from person to person. Roughly it is 25 centimeter in length. This esophagus will connect mouth with the stomach. Now, important point is it does not contain any enzyme. That means no digestion is taking place in esophagus or the food pipe. So what is it that happens to the food or to the bolus while it's traveling all along esophagus? Peristalsis. Now, what is peristalsis? Peristalsis is a rhythmic movement or rhythmic contraction and expansion. Contraction and expansion of the esophagus walls. That actually facilitates, or it helps the food, this bolus, to move down or to push it forward. Now, the part of the esophagus that is pushing the food, here you can see the esophagus will contract, and the part of it which is receiving it, it will expand to it. So contraction pushes the bolus downward or forward, and expansion receives it. That's how it travels all along the length, and then it is ready to enter one of the important part of the alimentary canal, which is stomach. This is stomach, all right? It has glands, and those glands will secrete certain juices containing some of the important enzymes and other important constituents. Now, for every organ in biology, there is a root word. So for stomach, it is gastro. So we can say the glands that are present in stomach, gastric glands. The juice secreted by gastric glands, gastric juice. Hence, you will find that next to stomach, I have written gastric juice. Right, now this gastric juice contains HCl, it secretes HCl, which is very strong mineral acid, right? HCl, then second pepsinogen, which is inactive form of the proteolytic enzyme. Proteolytic, proteo means protein, lytic means the enzyme that is going to act on lysis or hydrolysis of protein. So this is an inactive form. Now, what else is mucus? Now, these are the three important parts or constituents of the gastric juice. Now, let's see, what do they do? What do they actually perform? This HCl, being very strong acid, it brings about the acidic environment, highly acidic environment, approximately 1 to 2 pH. Why such acidic environment is necessary? So that this inactive pepsinogen can be converted into active proteolytic enzyme, which is pepsin, and then this pepsin can start hydrolyzing or digestion of proteins. So this pepsinogen, with the help of HCl, by creating such acidic environment, 1 to 2 pH, will be converted into active proteolytic enzyme, which is pepsin. This pepsin, as you can see, will act on proteins and will convert it into simpler polypeptides, which are peptones and proteoses. It is fine if you do not remember the names. I know the names are tricky, peptones or proteoses. Or you can simply write proteins are broken down into smaller polypeptides. But if you really want to impress the examiner, I suggest that you should write peptones and proteoses. Right. Why is mucus necessary? If you know, HCl is a very strong acid. It can damage the inner lining of the stomach. So there has to be something that can counteract that, right? So that part is mucus. This mucus will line the inner lining of the stomach and will prevent the damage that may cause by the corrosive action of this strong acid, which is hydrochloric acid. Then this, along with the chemical digestion, the food is also mechanically churned inside the stomach because it's a highly muscular organ. So mechanical churning along with the chemical digestion is all happening in the stomach. Then this food, semi-digested, the digestion is not completed yet, is ready to enter the small intestine. As you can see, small intestine. What's so unique about small intestine? It is large. It is approximately 6 meters long. You know, that is why it is all coiled up inside the large intestine. It fits inside the large intestine, right? That's why, being so large, it is divided into 3 parts: duodenum, jejunum, and ileum. So the food that will exit the stomach is given the term chyme. Earlier it was bolus when it was exiting the buccal cavity and was entering the esophagus. Now the food is coming from the acidic environment. It is leaving the stomach and entering the first part of the small intestine, which is, you're right, duodenum, right? And it is referred to as chyme. Now, before we discuss more about enzymes, let me tell you, the enzymes are very particular, very specific of the conditions or the environment in which they work. For if pepsin required highly acidic environment, if it is not given, it won't work, it won't catalyze the digestion of proteins. Likewise, if other enzymes require alkaline medium, then they need to have alkaline medium, otherwise they won't work, right? So when the chyme enters the first part of the small intestine, duodenum, along with that, duodenum receives 2 secretions, right? One from the liver, that is the bile juice. Another from pancreas, the pancreatic juice, right? Now the liver, as you can see, secretes the bile juice. Bile juice does not contain any enzymes. That means no chemical digestion it is responsible for. So then what is its significance? Significance is it brings alkaline medium, which is very necessary for the working of pancreatic enzymes over here, right? What else? It helps in emulsification of fats. Now, what is emulsification? Emulsification is breaking down of larger fat globules into smaller ones. So that increases the surface area and helps the lipolytic enzyme to act on it and catalyze the digestion efficiently, right? These are the 2 important functions performed by bile juice, which is secreted by liver and it is stored in gallbladder. Can you see this globular structure over here? That is gallbladder. It stores bile, it concentrates it. Then from pancreas secretes pancreatic juice. The pancreatic juice contains very important enzymes that I have already listed over here. It will contain pancreatic amylase, trypsin, pancreatic lipase. Now this P stands for pancreatic because amylase was also secreted by the salivary glands. So there it was salivary amylase, here it is pancreatic amylase. Now pancreatic amylase will act on starch and will break it down into maltose and simpler sugars, right? Then next, what will happen to peptones and proteoses, right? Now these peptones and proteoses will be acted upon the proteolytic enzyme of the pancreatic juice, which is trypsin. So trypsin will convert them into peptides. Still the digestion is not complete yet. Now, the lipolytic enzyme, this pancreatic lipase, will act on fat that is already emulsified by bile juice secreted by liver. Yes. So this emulsified fat will be acted upon by pancreatic lipase and will be broken down into fatty acids and glycerol, right? So this is all happening in the duodenum, right? This all digestion is happening here. But as I told you, the digestion is not yet complete. It still has to happen because I'm only talking about small intestine and that also duodenum. What happens next? Because I told you digestion is not complete. For that, you need to watch another video, right? Now, if you found the content useful and if you feel that I have somehow managed to make this human digestive system one of the difficult topic of biology simple, easy and interesting, please like, share and subscribe to my channel. That support is really necessary and that will be very encouraging for me. Till then, take very good care of yourselves and I'll be looking forward to all of you watching the next video that will complete the human digestive system, and I'm going to show you by the end of it, you will not have any question, any doubt left. However, still, you know what to do. You can always mention it in the comment section and I will be at your service. Till then, take very good care of yourselves. Stay happy, stay healthy.